Today, I'd like to introduce Dr. Louis Scheffer. As a fellow of Howard Hughes Medical Institute, working at Janelia Research Campus, Dr. Scheffer is focused on reverse engineering of the brain and applications of electronics to biology. For more than 2 decades, Dr. Scheffer had been working to improve the design of microchips, figuring out how to connect hundreds of thousands of electronic transistors in a single device. At Janelia, he's now applying similar design principles to understand another type of wiring, the intricate interconnections that neurons make in the brain. In particular, he wants to teach computers to recognize different types of neurons and axons and dendrites connected to them. So before joining Janelia, Dr. Scheffer spent time as a fellow at Valid Logic Systems and at Cadence Design Systems. He was also the WACI chair of the Design Automation Conference, and Dr. Scheffer received a BS and MS in electrical engineering from Caltech and a PhD in electrical engineering from Stanford. And so, Dr. Scheffer, I was wondering if you could tell us a little bit more about your story.
It seems like a big switch from EE to biology, but it actually happened in a series of relatively small steps. When I first got out of school, I was pure EE, and my first task was to design a circuit, build a circuit on a chip for Hewlett-Packard, which at that time was an instrument company. And it was small, a few thousand gates, and was designed by hand, as people designed the first chips. And it took about 2 years, so it took quite a while. And when I was done, even at that time, you could see that chips were getting bigger. And so when it got bigger, you would need more tools, and different tools. You couldn't design it by hand anymore. And so I switched into the software field, where we built software to help design chips. And that's where I was for the next 20 years, perhaps 30 years, at Valid and Cadence. That every year the chips would get bigger. Every year we would try to improve the software that you use to build the chips. And we were able to keep the time to build a chip to about 2 years, even though the team got bigger and bigger each time. And so by maybe early 2000s, the chips had hundreds of millions of parts. The teams were maybe 50 designers, and it still took 2 years to design a chip. And so what happened at this point is because you have 50 people working for 2 years, the chips cost maybe $10 million or more to design, just do the design, not even manufacturing. And so people started to consider whether or not designing such a chip is worth it. And to do that, you have to guess how much it costs. And in order to guess how much it costs, you have to know how big it is, because that determines how many you get on a wafer and the odds of having a bad spot in the chip that'll destroy the function. And so the next step was to figure out, before you build the chip, how big it's going to be. Now you can simply add up all the stuff that needs to go in. Say it needs this many multipliers and this many adders and this much memory. You could just add that up. But you also have the wiring that connects all the parts. And just like you couldn't specify a city from scratch by just adding up the area of the houses and work buildings and things, because you need to put in the roads. And so you need to keep room for this interconnection between the different parts. And that's going to be one of the main determinants of your cost. And so this subfield sprung up called interconnect prediction, where given a chip and a description of what it's supposed to do, you try to figure out how big the wires are going to be and how much area they're going to take up. And so it was a specialist subfield. There were maybe 50 people in the world worried about it. And in the early 2000s, I was organizing a conference to talk about this. And one of the people on the conference committee said, hey, do you know there's a biologist who's looking at a really similar question in a small animal called C. elegans? It's a little tiny worm. But he was figuring out in that worm how much area was occupied by the neurons and how they talk to each other. And we said, hey, this sounds pretty cool. So we invited him to the EE conference. He talked about it, and everyone thought it was interesting, and invited him to a few more conferences after that. And about 2 years later, he called up and he said, Howard Hughes Medical Institute, which is this big research organization funded by Howard Hughes's money. He never had kids, so it all went into research, was building a new research campus. And would I like to come out and give a talk? And so I said, okay, I'll come out and I'll talk about how EEs approach the same problem. So I did that. And I went out. There were a lot of interesting people, a lot of interesting problems and things. And then it turned out that the Howard Hughes Medical had a visiting scientist position where you could come just for 9 months was the plan and work on a problem. And so I took a leave of absence from the electronics industry and went there intending to stay 9 months, do some work and then go back. But this was 2008. And so there was a big industrial downturn in the Silicon Valley side of things. Plus, having spent there 9 months, it was actually really interesting problems and things. And so at that point, because Howard Hughes was self-funded, it didn't have the budget problems that industry had in 2008. And so they still had open positions. And so I applied for one. And that's pretty much how I got connected into biology. How I got into reconstruction and connectomics there is I thought the first thing to do when I got to Janelia, which is the campus of the Howard Hughes Institute, would be to study these circuits and try to figure out, use double E techniques and figure out how they work. But it turned out there was only one animal for which the circuit was known, which is this tiny worm, C. elegans, that has exactly 302 neurons. And so the first step is to try to find more circuits. And so that involves reconstruction. And then reconstruction involves all sorts of technical problems of imaging and alignment and dividing images into neurons and everything. And so that's how I got into reconstruction.
Yeah, it sounds like it's been a really exciting journey. You know, you've got to explore lots of different fields or subspaces along the way. And we were talking a little bit before the episode, and you mentioned over the long term, it looks like a big, kind of like a huge jump from where you started to where you ended up. But as you were kind of explaining, you really did it one step at a time. And yeah, I think that it really resonated with me how like each step in the process was kind of at least somewhat connected to the step before, and a lot of parallels between those fields that you've got a chance to be involved in. And, you know, I'm sure we'll go into some of the details throughout our discussion today. But before we dive into the specifics, I was wondering, when did you notice that first initial spark of interest in the neuroscience space? Did it first happen when you watched that talk by that professor at the conference, or were you always interested in neuroscience a little bit even before that?
No, it was pretty much out of the blue. And it didn't even happen at the talk. It happened at the breakfast before the talk, because the guy who was coming, he wasn't familiar with double E. And he said, all right, what's the audience going to be like? What kind of words should I use in talking to them so that they'll understand what I'm talking about, and so on. And so over breakfast, he was describing what he was doing, and I was describing what we were doing. And that's when I first started to get interested. Then he gave the talk. That was good. But it was really that one breakfast conversation that was the actual starting place.
It's awesome how something so exciting as the like this pivot that you were able to work on, or that you were able to accomplish, really all started out of this breakfast conversation.
Yeah, and it was, if you look backwards, even before that, it was probably when the colleague on the steering committee, which I think was Igor, I'm not sure who it was, recommended this guy. Right, so that was really the step that started the step that started the step. So it's I know it's good. And how he knew of it, I don't know. And so it's a rather indirect story, but it was something that was easy to remember.
I'm glad that you guys ended up crossing paths and came into contact.
I know one of the things you wanted to ask about was what is connectomics? And that's a sort of an interesting question. If you look on Google Scholar, you'll find two completely different branches of study which are called connectomics. There are people who are at the top scale who are using fMRI to look at the brain and try to figure out where the connections are between the brain. And this is at a very, very high level. Like their analyses, if they can get down to the millimeter level, they think they're doing really well, a cubic millimeter of the brain. And they're also somewhat controversial, because when you have that many pixels in the brain, practically for any task you do, you'll get some that stand out. And so people always wonder how reproducible the data is and so on. And there was a really famous paper in this area where some guy took a dead fish and put it in the scanner and was able to reproduce some of the results. Reproducing results from a study that showed people pictures and asked about their emotions that as seen in the picture. And so he did the same experiment with the dead fish. The fish is in the scanner, he's showing it pictures of people with different expressions on their faces and running the MRI on the fish. And he got a number of positives. And so he used that as an example of the fact that when you have this much data, you're going to get false positives. And so you really can't, without a lot of statistical work, necessarily take this work at face value.
Yeah, I think I heard of that dead fish experiment a couple of times before, and it really does get his point across when talking about those false positives there.
Yeah, it's pretty silly, but it does get the point across, as you said. Our connectomics is a little bit different. It's quite a bit different, actually. Like every pixel they have is, like I said, it's a millimeter on a side. Our pixels are 10 nanometers on a side, which means that every pixel that they have, we divide into a million billion pixels. And so it's a cube, 10 to the 15 pixels of our pixels are inside their pixel. And so we try to look at the actual cells, the neurons in the brain, and the connections between them. And the connections we see are the chemical synapses. This isn't all there is, but it's a good fraction of what there is. And so a connectome, in our terminology, is a list of neurons and a list of the connections between them and everything we know about those connections. And so, as I mentioned, it was only available for these tiny creatures before, C. elegans, 302 neurons. After 15 years of work or so, we're up to the region where we can do a fruit fly. And a fruit fly is this millimeter size animal. And even so, in the brain, it has 150,000 neurons with about 150 million connections between them. And so even that has been a struggle to try to generate. But if you go online and you look at, there's a website and everything where you can now go in and look at those neurons, call out the neurons by name. They're all named through efforts of many zoologist classifiers, and you can look at the connections between them. And that's what we currently call a connectome.
Yeah, it seems like you and your team are much more zoomed in than that other subspace of the field. And I'll definitely have to go on and check out the fruit fly connectome website that you mentioned. I can imagine that moving up from just 302 neurons to over 100,000 neurons, the complexity can just be going up astronomically as to analyzing the connections and such here. And so this kind of drives me towards my next question. As we dive into a bit further detail, your background in chip design is pretty well suited for projects of this scale. And in both domains, there's large groups of people working to solve a common problem. There's, as you mentioned, millions of components. There's lots of factors at play, more than just algorithms. And so I'd love to hear you expand a little bit further on kind of how some of the similarities you may have seen between like microchip design and analysis versus what you work on in your day-to-day now and kind of your vision for these current projects.
Yeah, sure. There's the microchip design really hasn't had that much effect, except for the fact that you're not scared of dealing something with millions of parts in it. Because we can't design nervous systems yet. And so a lot of the tools you have, synthesis tools and simulation tools and things like that, we don't have for nervous systems at this scale. What is really relevant is there's something that's important in ICs, which is called reverse engineering, which is where you take your competitor's chip and you pry the top off, and then you look at it with electron microscopes and you figure out how it works. So you can try to figure out what its strengths and weaknesses are. And that is very similar to what we're doing, except that we understand the technology a little bit better in the chips. But you need electron microscopes, you're analyzing pictures, automation is super helpful. It's actually a pretty close parallel in that respect. It's also important because that's where we get all our technology from. If it was only biologists, no one would build these electron microscopes and stuff that we're doing. They get there because of the semiconductor industry. The semiconductor industry has to examine these big things like mass and stuff to get the details. And so they are the ones that pay for the development of the tools that we then co-opt for biology. And so we're very lucky that they're in a similar situation of having to do a lot of looking at very small parts, because we could not afford to have funded the development of those things ourselves.
Yeah, it makes sense that these fields kind of go hand in hand. And as you mentioned, there's a lot of money pumping into and around of the semiconductor industry. And it's definitely good to hear that, a lot of the research and development for these types of tools that they're building have been able to be applied to things like biology, where maybe there's not as much money or funding coming into these specific aspects of the field. And so one of the projects that you're working on was looking to rebuild reconstruction infrastructure. And I was doing some reading on your lab website, and it mentioned that the legacy reconstruction decisions and maybe what the current technology is using is a binary choice, and that would lead to significant data loss. And so your lab was looking to basically turn this into a probabilistic process at a variety of different levels, both at the neuron level and then even just raising up in complexity to the brain region and even the structure within that brain region. I was wondering if you could explain to our listeners some of the key benefits to changing this reconstruction process over to a probabilistic framework, but then also what are some of the key obstacles that are getting in the way of this, or that at least must be crossed before before things can be looked at in a probabilistic manner?
The probabilistic work is actually important at a whole bunch of levels. And let's say at the very lowest level, you're deciding whether this group of pixels is connected to this group of pixels over here. And originally, we would just make that decision one way or the other, a binary choice, like you said. But now we have a probability. What's the odds that these two are connected? And that turns out to be important because now you can adjust your algorithm depending on what you're doing. So for example, if you're too aggressive about combining things together, you accept a lower probability of junctions. What you get is these big things where neurons are merged together, makes a big mess, and they're extremely hard to split apart. On the other hand, if you're too conservative, what you end up with is tens of thousands of tiny fragments, now all of which have to be put together by hand. And so by making it probability, you can draw that curve and decide where is the best point to sit along that curve. And for us, for example, we made some measurements of how people were doing and said, oh, it's about 6 times harder to split something apart than it is to combine two things that should have been combined. And so where we should be on that curve, we should pick a point where now we spend equal effort splitting and merging, which means we have 1/6 as many places where we have to split as we have to merge. And then the same thing holds at the next level up. Right now you're talking about, say, neurons. Well, depending on your analysis, you may want only the connections that you're really sure of. Or on the other hand, you may want as many connections as possible. Maybe you're doing a statistical test. And so that's something, again, where you can use these probabilities to say, okay, I'm willing to take, as long as it's at least 70% certain, I'll take it. Versus the other analysis where you say, nope, unless it's 99% sure, I don't want it. And so probability affects the things at every level. Now, one thing it doesn't affect much is this brain region thing. It turns out if you look at flies, at least for flies, the detailed wiring is very, very different on each fly, one fly to another. It's not like a circuit board or something where 2 of them have exactly the same wiring. But the regions are the same. They'll still have this region for vision and this region for smell and this region for controlling the nerve cord and so on. And so the regions are super stereotyped, but the circuitry isn't. And so that's one of the areas. Now, even in the circuitry, sometimes it's hard to explain. Sometimes it's sort of the same, but not exactly. So if one neuron has 50 synapses between 2 neurons, the next one over might have 45, and the one next to it may have 70, and so on. They're statistically not all that well controlled, but the averages are pretty good. On the other hand, there are some circuits where, like in the sense of smell, where it seems to be wired explicitly randomly. If you're familiar with computer science, you're familiar with the idea of hash tables, which take advantage of randomness. It seems that some parts of the fly brain have done that as well. And in those parts of the circuits, even if you look at 2 sides of the same fly, they're completely different because they were chosen pretty much at random. And so this idea of randomness seems to be embedded in the circuits all the way from the bottom to the top. And one thing that also we're studying, which is interesting, is that the circuits may be random when they're built, but they may also be tuned. We know that a fly, when it's growing up, it's in the pupa. It just grew from a larva. It's got eyes. The eyes are not open, or they're not seeing anything because they're inside the egg case. While it's in there, it develops something called patterns that go across the retina, the equivalent of the retina, bars and stripes and things like that. And what that may be doing, we're not sure, is helping to tune the connections that exist that were formed genetically to make the fly optimized to be able to see motion and see other things that it might be seeing. And so it may be that even though the initial wiring is sort of random, the operation is not as random as the wiring would indicate, because these other things like this pattern stimulus may correct for some of the differences that we think are random. And so it's a fascinating topic to see how these things try to work.
Yeah, that's super interesting that we're seeing, as you mentioned, things like randomness and perhaps even tuning going on in the fly brain, and how we're seeing, certain processes that might even contribute to this tuning. And one thing that popped into my head as you were giving that explanation there is you mentioned that the regions are generally connected in a similar way across basically all of the flies of the species, but the actual neurons are connected in a much more random or just not necessarily in the same way. And I was wondering, if we go to a simpler organism like the C. elegans, are we kind of finding the same thing? Or in the C. elegans, are the neurons themselves kind of behaving more like region?
They're more like the regions. They're very stereotyped. Part of it, I think, is because with only 302 of them, everyone is unique. It has its own genetic code. It has its own targets and things like that. Whereas in the fly, a lot of it is determined by growth. That if you look at how many of these cells a fly has to look at odors, to understand odors. In a well-fed fly, you might have several thousand. In a poorly fed fly, you might only have 1,000. And so probably what it did is it grew until it couldn't grow anymore, ran out of nutrients or ran out of whatever, ran out of time, and then built the circuit with what it had. And so, but the little tiny worm can't do that, I don't think. And so I think the fly is built differently. It's not as stereotyped as the worm.
Yeah, that makes sense. We're not necessarily seeing the same level of redundancy in the worm as we are in the fly. So we're not seeing extra cells based on nutrition, for example, that might feed into the same activities as some of those stereotype cells, basically.
Yeah, it's sort of the whole idea of how biology builds things is sort of fascinating. I think that's another huge question, which is how does the brain get built, as opposed to how does it work when it's an adult, and how does it decide how many cells to build and whatever. And that process is clearly very forgiving, because if you look at the higher level things, like some people are born with 6 fingers, and if you look at that extra finger, all the nerves are right. It's got muscles. It's got everything. So clearly the process allows quite a bit of variation, right? Just because you have 6 fingers, you won't have 5 nerves. You'll have 6 nerves and everything to go along with that. And so the process of building has to be pretty tricky. And it's something we don't understand yet.
Yeah, absolutely. Definitely something that we're still exploring and still looking into. But this does kind of segue into my next question pretty well. I was hoping to talk about next another goal that your lab has, and that's to incorporate biological knowledge into these reconstruction models and techniques. And so I was wondering if you could expand a little bit on some of the ways that your team is working to bring this biological knowledge into reconstruction from an early stage, and kind of keep this biological structure and process in mind all throughout the reconstruction phase.
Yeah, sure. There's a really easy example to use, which is that in the brain they use maybe 7 or 8 different neurotransmitters, chemicals that transfer out of one cell and affect the next cell to send messages. And in general, each cell uses only one of these. There are some exceptions, but in general, each cell only uses one. And so, say if you're looking at 2 fragments and you're trying to reconstruct it, and you say, should these be joined together? Then, if you look in this half and say, oh, this uses acetylcholine, and you look in this half and say, oh, this part uses dopamine, then you're pretty sure that those 2 do not connect together, even though they might look like it. And that would be an example of biological information that helps you do your reconstruction. Now, that particular one is extremely interesting, because if you ask the human, what's the transmitter used in this part and what's the transmitter used in that part, the human can't tell you. The synapses look the same. The EM synapses look the same to us as people. But it turns out if you take machine learning and you say, okay, here's 10 million acetylcholine synapses, here's 10 million dopamine synapses. Now here's a random synapse. Tell me what kind it is. It can actually do that with somewhere better than 90% probability. And if you ask it, you try to ask a neural network what it's looking at, they'll tell you, oh, I'm sort of looking at the whole thing. Right? So it's really sort of a mystery. But yet the machine can tell you quite easy or quite reliably which neurotransmitter it's using. And then once you have that information, you can now use that to decide whether to make these merge or split decisions. Likewise, if the opposite case, which is where you have a single thing, but you see 2 neurotransmitters, probably you made them mistakenly merged, and you probably ought to split those 2. And so that's one example. A more higher level example is what the human annotators who are looking at these fragments do, which is they have a good idea of what a cell looks like as a whole. They say, all right, this cell normally comes from this region over here, goes to that region over here, typically has 3 or 4 branches in this area, has little feathery looking tufts on the end, etc. And they can make decisions that right now the neural networks and things cannot, or whether this looks like a sensible cell or not. And that's biological information. It's on the shape, but it's something we don't have a good representation for in the machine. And so those are 2 examples of sort of biological differences that we can tell. Similar, a few more might be if you look in this half and there's lots of small dark vesicles, and this half doesn't have them, or doesn't have the same microtubules or whatever, it's much more likely that they really aren't connected. So the closer you look, the more things you can see, the more likely you should be. You should think that if 2 things are nearby and all their details match, they're probably connected. If they don't match, they're almost surely not connected. And so there's lots of ways to try to get this biological information in there.
It's really interesting that some of these pieces of biological knowledge that you gave as your example are easier for the machine to distinguish between, and then other ones are easier for the humans to distinguish between.
Yeah, that's something we're still working on that. There doesn't seem to be any theoretical reason why that should be so. Maybe humans could be trained to tell the difference between these synapses, and maybe machines could be taught to learn how to represent these shapes and things like that. But at the moment, there is that divide that you just mentioned. Some things are easier for the machine, some things are easier for the person.
Have you found any factors that seem to influence which tasks might be easier for the humans versus which might be easier for the machine learning techniques?
The ones that sort of involve these subtle variations, where the synapse looks almost exactly the same, but the cleft is 2 pixels wider or something like that, the machines seem to be better at picking that up than people. These ones that deal with shapes, machines are not very good at shapes yet. So if a person can look at a tree, there are directories online where you can just look at the shape of a tree and tell what it is. People are very good at that. And people also have a extremely good memory for shapes. Some of the best people have been working on this stuff for 10 or 15 years, and they'll just look at a neuron and say, oh, that's an MI1. That's an L4. That's whatever. They can classify them almost as quickly as they appear on the screen. And that's something that the computers are not good at yet. Now, maybe they will get that good because right now, of course, they're being trained on images of roads and people and trees and stuff, not on images of neurons. Maybe in the future, once we have enough neurons, then we can train the machines, and the machines will become as good as the humans. But they're certainly not there yet.
Yeah, that makes sense. The machines need more data, more images to really start to learn these shapes better. And those humans that you mentioned that are able to really just glance at a neuron and know what kind it is, that's super impressive.
Yeah, it's super impressive and it's super valuable. And these people are rare, both because they have to have the natural talent and then they have to have studied for a long time. They're probably one of the resources in our reconstructions and things that would be most difficult to replace. Yeah, you can get another electron microscope expert somewhere. You can hire software people, do these things, but this kind of biological knowledge is hard won, and it's super valuable.
And so we've been talking a lot about incorporating biological knowledge into the process and at lots of different levels. And I was wondering if we kind of zoom out and look at the field at a large scale, what are some of the efficiencies or doors and opportunities that this can open looking forward?
There's a bunch of things you can see happening in the short term. One is that right now, the connectomes are a little bit limited, which is that you can't look at a connectome and say, this is how the circuit works. And we actually have some proof of that because Srini Taraga, one of the guys in the Genelia labs here, tried an interesting experiment, which is he took the connectome for the optic lobe in the fly. And then what he did is rather than have a neural network where everything can connect to everything and it learns whatever needs to be connected, we're going to start with this one that we know exists in the fly, and we're going to train it to do a task, which is detecting motion, that we know that this network does in the fly. And what he found out is that the solution is not unique. There were many different combinations of synapse weights and things like that that could give the same function, even if you're restricted to only having the neurons that are known to be connected in the connectome. And what that tells you is that you need to know, to figure out which one it actually is, you need to know more. You need to know maybe which way these transmitters are, because some are inhibitory and some are excitatory. And he found if he got that data out of the biologist, then the connectome would only converge to one possible solution. And so, but that's an example of what we need. It's the kind of information we need. And that's one of the things that's in process now. People are looking at how they can get this additional information that's not in the connectomes today. There's 3 or 4 kinds of that. One is there's something called gap junctions, which is normally in a synapse, a neuron A gives off a chemical that's received by neuron B that causes some effect in B. But they can also actually put their membranes together and form a little hole in the middle and just exchange charge, just like jumping a car or something like that. So you can handle it directly. And it turns out in the electron microscope preparations that we use for connectomes, those are hard to see. You can see them if you take super high mag and other things, but you can't see them in the images we have. And so that's missing from our connectomes. The other thing that's missing is something which are normally called neuromodulators. And these are ways that neurons can talk to neurons that are not directly adjacent. What they do is they take some chemical like adrenaline is a typical example in a person, and one cell gives it off, but instead of just affecting the next cell, it soaks through the region in between the cells and can affect the cells in a wide range, even without a direct connection. And that's also missing from our current connectomes. And in that area, we have to understand both where it's given off and where are the receptors on other cells that can receive that signal. And so there's, in the near term, maybe 3 or 4 years, that's something that has to get added and processed and handled in the simulations and things like that. So there's a lot of stuff that's needed that we don't have yet. And then in the longer run, there are some technologies that are sort of coming online. I don't know if you've heard of something called expansion microscopy. Have you heard of that?
No, I haven't heard of that.
Yeah. That's a really weird thing. It's one of these technologies I would have sworn up and down is never going to work. But what they do is they take something like a cell and they embed it in a plastic that's like the plastic they use in disposable diapers that expands when it's wet. And then they apply some chemicals that break the strongest bonds in the cell. Then they dump in water and the whole thing physically expands. And so if you can do that enough, you can get to the point where you can see in optical microscopes what you could only see before in electron microscopes. Now, people were pretty skeptical about this. It doesn't seem like the kind of thing that was going to expand uniformly and everything else, but it seems to work. And so modern people have got that now. Recently, there was a paper reported where they successfully expanded the sample by 16 times. And so if you make it 16 times bigger in every direction, now with your optical microscope that can see 300 nanometer objects, that you're looking at objects that started out at 20 nanometers, which is about the size you need to see a neuron. And so maybe if you can make this work, you could use optical microscopes where we need electron microscopes now. And that would be a huge plus because electron microscopes are big, they're expensive, they're finicky, they're hard to run. But optical microscopes are something you can buy off the shelf. Not the fancy ones they need, but you can more or less get them. And so that's another possibility for something that might happen. I would say maybe 5 years out for that, assuming they can get it to work.
Yeah, that's incredible. Like instead of focusing on zooming in further, they're just deciding to make the thing they're analyzing bigger.
Yeah. I would, like I said, I would have, if I was in a funding agency and someone had asked for funds for this, I would say that's such a long shot. It's not worth even trying. But I'm glad it wasn't me who made that decision because it's looking like it might be successful.
Yeah, that's really impressive.
Yeah. And there are also other techniques that might get connectomes that don't involve microscopes at all. For example, one is referred to as barcoding, which is the idea is maybe you can, through genetic techniques, get each neuron to generate its own little unique genetic tag. And then you can introduce things or some things like rabies viruses that are known to travel through synapses, but not through cell walls. And so now if you have two different tags in two different cells, and you can get them to go through the synapse, you can generate little pieces of DNA or RNA that have both tags together, concatenated together. Then you can smash the sample up into a glob, run it through a sequencing machine, and look at all the sequences you get out. And that will tell you exactly which one of these cells were connected to which other one. And so you could generate a connectome without ever looking at pictures at all. Now, it has some obvious disadvantages, which is you don't know physically what the cells look like. That might have been important and so on. But at least you could get a basic connectome with no imaging required at all. That's another possibility that you could do. There's a third one, which I don't think anyone has quite got working yet. You can use these big specialized facilities with synchrotrons that generate really, really short wavelength X-rays. And with those, you can examine a volume in a much shorter time than it takes us with the electron microscope because we have to do it a layer at a time. They can do the sort of volumetric thing by just X-raying the sample. And so there's all, a number of things that may help the connectome field out. we'll have to see how those go. But I think the future is pretty promising.
Yeah, it sounds like a bright future ahead, and lots of really interesting and useful techniques that we might start to see soon here. And I think something you mentioned is none of these techniques are perfect, but who knows, maybe if we combine them in the right way, we can get certain pieces of information we need from each one.
Yeah, that makes a lot of sense. In something like the fly anyway, where their circuits are more or less the same in every animal. So if you have one technique that can tell you where the neuromodulators are given off and which cells receive them, and then you have the synapses, which you get from the electron microscope, and then maybe you get the gap junctions by some third technique, you can combine them all in one model. And so the fly is very good for that. Whether or not that would work in a bigger animal, like a mouse or something, is not at all clear, at least yet.
Yeah, so it's hard to tell if it will translate up to organisms that are a bit more complex.
Right, yeah. There's two other things that I think are coming up relatively soon that look pretty interesting. One is switching from flies to larvae zebrafish. And the flies are great because we have really good genetic control of all the nerves and things. So we can test the hypothesis pretty easily by triggering a certain nerve or deactivating it or whatever. But the zebrafish has a completely different advantage, which is it's transparent. And so the zebrafish larva, that's its defense mechanism, is to be transparent. So it's hard to see. And so what you can do in the larva is you can induce in each cell a optical reporter, which is a dye that glows when that particular cell activates. And now what you can do is you can train the fish, you can image it with an optical microscopes, and you actually watch the neurons go on and off and flash as the fish thinks about the problems it's trying to solve. But then you can take that same fish that you already trained and feed that to the EM. And so you have not only the connectome, but you know what that particular fish had learned. And so that's something we cannot do in a fly, right? We have to get the flies when they're pretty young. Flies are hard to train anyway. Even a really good fly only does the thing you want 70% of the time. And so this working on the fish may give us a lot more information, particularly about learning and things, than we can get from the fly.
Yeah, absolutely. It seems like a lot of behavioral types of things can be discovered from a situation like that, as well as you mentioned, just the learning going on within the brain.
Yeah. There's another thing that's going on, which isn't certain yet, but people are working on it, which is maybe move up to the bee. The bee is harder technically because it's brain is quite a bit bigger than a fly, but it can do a lot more impressive behaviors. In particular, it's well known that bees can return to the hive and tell their buddies where a flower is. And that's something a fly could never, as far as we know, couldn't do. And another one that to me is spectacular is a bee that watches another bee do a task, in turn does it better itself. And so this is pretty wild because what you see, of course, is another animal. It's reversed and everything. So even just trying to do what another animal did, that's a very non-trivial problem, sense of self and non-self and directions and everything like that. But it's been shown that bees can do that. They give a bee a nonsense task like rolling this ball to the middle of the table, and then they feed it. And the next bee you train, if it can watch the first bee, learns it faster. And so that to me is much closer to what we'd like to really understand about brains and how they work. And it's only maybe 10 times bigger than the fly. It's not nearly so big as a mouse. A mouse is 10,000 times bigger than a fly.
Right.
Studying it in mice is hard, but a bee might be a really good compromise. It can do a lot of clever tasks, and it's not so big as to be completely impractical with current technology. And so that's another good thing that may come out in the next 4 or 5 years.
Yeah, that's really incredible. I didn't even know a lot of these learning capabilities that you mentioned about bees. And yeah, that's really incredible how it's not that much bigger than the fly, but can do some of these really impressive things that are likely neural based. And do we have pretty decent control over the genetics of a bee like we may over a fly?
No, I don't think so. I know a fly is far and above anything else. It's been worked on for a century or so for genetics. I don't think we have it for bees. I think in fact that's a point of contention in the field, is that honeybees are known to do a lot of these things, and they're small, but maintaining a breed of honeybees is hard because sort of the genetic unit is a hive. And so it's expensive and difficult to maintain genetic strains of honeybees. Whereas I think bumblebees live in smaller groups. They're easier to handle genetically, but I think they're also bigger. The brains are also bigger. And so I know that the bee people are worrying about this kind of detail now. They're trying to organize an effort to do some sort of analysis on bees. So don't know the details myself, but it seems promising.
Well, definitely interesting thing to keep an eye on over the next few years, sounds like.
Yeah. And then I think there's in the medium term, what we'll see is some of these other animals, the fish, maybe the zebrafish, maybe the bee done, maybe in 10 years, we might have portions of a mouse brain, just the hippocampus or something. Although, quite frankly, in the fly, before we did the whole fly, we also did little pieces of the brain. And they were always hard to interpret because you don't know what those neurons are doing that are sticking in from the outside. That made it hard to understand what they were about.
Yeah, it seems like that can make things complicated for sure.
Right. But doing a whole mouse is quite a deal, right? If it took, $40 million or so to get the technology and the people and stuff to do the fly, and it's 10,000 times bigger. That's many billions of dollars. And that's an enormous investment for someone to make. It doesn't make sense to make it yet anyway, because it's back earlier when you referred to the semiconductor industry and what crosses over from the semiconductor industry. One thing that crosses over, I think, is that incremental improvements over a period of time result in huge changes. Like the chips never got that much bigger in a given year, right? Every year and a half, every 2 years, they get twice as big. But you do that 10 times over the course of 15 years or so. Now you're doing things that are 1,000 times bigger than before. And so I think that's probably the right way to approach the mouse is don't just start working on the mouse, work on the technology. And if you can keep improving that technology by, a factor of 2 every 2 years or so, then maybe in 10 or 15 years, be ready to work on the mouse.
Yeah, that makes sense. It's like the technology needs to progress a little bit before it's ready to take on this challenge of the mouse.
We've looked at it. We said, what would it take to do a mouse? It would just run into some pretty difficult barriers at the moment. Like the machine that we're using to take the electron microscope images, we've got one of the fanciest ones available. They're $5 million each. You just work your way through and you say, how many would we need to do a mouse brain in less than, say, 2 years? And you need 1,000 of them, right? That would be, $5 billion. Plus, I'm sure if you ordered it, they couldn't deliver it, right? These things are built to order. They probably consider the semiconductor industry as their biggest sales, and maybe they sell 100 of them.
Mm-hmm.
If you ordered 10 times their capacity, they would have to build up the factory and everything. So it's just not the right time to tackle that, in my opinion. Work on the technology, and then maybe in 10 or 15 years or so will be the right time to tackle the mouse.
Yeah, that makes sense. And then past the mouse, is there any possibility to ever understand the human brain at this level, or would that be a long ways away?
That's a really good question. And I don't know. I think it depends a lot on the theoretical studies, because that in some ways the human brain is not that different from a mouse brain, because it's built using the same number of genes. Right? There's not a lot more genes in a human than a mouse. And so it must be in some ways a matter of building the same structure bigger. And so not in a completely different way. And so maybe if you did understand how a mouse worked, you could say, oh, okay, it's like a computer, right? If you have a small one, it can only analyze typing. You have a slightly bigger one, now it can analyze voice.
Mm-hmm.
You have a really big one, maybe in real time it can analyze video. But the computer is not really any different. It's just a difference in scale, what it can compute. And maybe the human brain is the same way. I don't know. Nobody knows as far as I can tell. But I think that kind of theory is what will eventually tell us whether we have a good chance of understanding the human brain. I'm personally sort of split about that, because there are some, I think, good counterexamples. For example, look at high temperature superconductivity. This is the one that works above cryogenic temperatures. People are pretty sure they know the laws of quantum mechanics. They know exactly how these materials are made. They can do any experiment they want, invasive, non-invasive, whatever. And they still don't understand, after 30 years, how these materials work. And it's just, a complicated problem. It's maybe it's past what people are capable of understanding. Just like you're not going to teach your dog relativity.
Right.
Maybe there's some problems that are tough enough that a human class thinker can't get all the way through it in a lifetime, and hence it never gets understood. Maybe that's a pessimistic view. Maybe these things are much easier to understand than that, and they'll figure them out. But I don't think it's a slam dunk either way, whether humans are capable of understanding how human brains work.
Yeah, and As you mentioned, we definitely have a long way to go if we are ever to get there before we do understand that. But I'm excited to kind of keep an eye on it and see what happens.
Yeah, it's fascinating stuff. But what the end thing is, we don't know. But I think for sure we'll understand the fly. A good chance we'll understand the mouse. Will we understand people? It's hard to say, but I won't be around at that point anyway, most likely. You also asked earlier about how to get into the field, if you wanted to get into the field.
Yeah, I was wondering, did you have any tips on breaking into the field, perhaps
Yeah.
for either students, young professionals, or maybe even somebody that's kind of mid-career and finds themselves getting interested in this space and kind of switch over after working on something else for a while like you did?
Yeah, I think it's I would say it's actually a pretty easy field to get into. It's not like theoretical physics or general relativity or something like that, where you have to study for tens of years to even get to where the state of the art is. So you could think of making some contribution. Neuroscience is still a very broad field that's not that well understood. And so like an average person can help out quite a bit. And I think there's lots of areas. For example, if you're a double E, electrical engineering, they're working on these probes that stick in the brain and all sorts of things like that. And if those projects are going on at like our university or our institute and universities and things, you can contribute there. There's a lot of computer science. If you're good at computer science and you want to do image recognition to try to figure out where these cells are in these images and how they're behaving, we're always looking for people in areas like that. Software engineering, these big projects require a lot of people to think about databases and storage and publishing the data on the web, but all these traditional software issues of getting the data out. The people who build these microscopes are all physicists, and better microscopes and things are always needed, improving the ones we've got. And there's a lot of room for chemists. The chemists build all the dyes and the indicators and things. Like I mentioned, the zebrafish, where you have the indicator that lights up when the cell fires. Those are all built by chemists. And so I don't know this for sure, but I would guess that our institute, which is a biology institute, probably roughly half the people have at least BS degree in a different area entirely, that all these people can contribute to biology pretty well. And there's also a bunch of room, I think, for sort of theory people. Right? There's a lot of theory that isn't well understood. For example, one that's also very practical is, suppose you have this system that looks at neurons and says which transmitter they use. But we don't have a good method for taking a system that's known to work and saying, how does it work? Why does it work? What is it doing internally, so we can teach people to do the same things that the system knows how to do? We don't have a good feel for that. We also don't have a good feel for, I don't have a good name for it, for what I would call somewhat nonlinear systems, which is you have a linear system of any size. People have really good tools for analyzing using matrices and eigenvalues and eigenvectors and stuff. All that's well understood. If you look at fully chaotic systems, it's pretty much hopeless. You can build a system with 3 variables that exhibits chaos, and it's really hard to predict anything. But biology is somewhere in the middle. The processes are quite nonlinear, but the thing doesn't go wildly out of control. And it can't, because if you look at a fly, for example, it's a cold-blooded animal. It has to work from maybe 10 degrees C to 35 degrees C. And during that temperature range, all the reactions are varying by factors of 3 or 4 and things like that. But yet the fly more or less does the same thing. And so, yes, it's a nonlinear system, but it's a nonlinear system that has a very well described and stable purpose and operation. And I don't think we have good math for that. And so if you're a theorist, you might want to go into that.
It does sound like there's lots of different ways to get into the field. I think you've mentioned chemistry, mathematics, theory, computer science, electroengineering.
Yeah.
There's lots of very interdisciplinary and lots of various different types of expertise.
Another one I just thought of, but it's quite big, is computer vision. Because all of these experiments with flies and fish and things have to be interpreted. And no one can stand there and watch the videotape and watch 1,000 fish learning a task and figure out what's in common between all these fish. And so a lot of the experiments now require a lot of computer vision. Track the flies, track the fish, see what they're doing and how they're doing it, whether they're learning their task and stuff. So that's another area you can get in. And so I would say, talk to your friends, see if you can find some friends in biology. There's a good chance that someone in your field is in biology now, and they maybe could give you some clues as to what's needed and where you could go, perhaps.
I think even from the aspect of engineering, even if you don't know that much about neuroscience, if you understand your field, you can start picking up the neuroscience knowledge as you go and figure out parallels or ways to apply it, it seems.
Right. Especially it's in the fields like the electron optics for imaging and stuff like that, or the people who build the probes that go in the brain. That's pretty hardcore electrical engineering. You're trying to build low noise amplifiers and things like that. But if you work in that group and you're hanging around biologists, pretty soon you'll say, well, what problem are you trying to solve anyway? What are you studying that needs this probe? And they'll be happy to explain. And so maybe in a while you might be doing your own experiments. Who knows?
Yeah, absolutely. You never know. And I've definitely found neuroscientists and biologists to generally be pretty excited and pretty happy to talk about, their research or what they are analyzing as long as you just reach out and ask a good question.
Yeah, they're pretty open so far. Yeah, I guess it's probably different in the startups and things. In the academic area and the research institutes and stuff, people are happy to talk about what they're working on.
To backpedal to something that you said earlier, I've been thinking about it a little bit. You mentioned that the gap junctions are really hard to see with the microscopy. And I was wondering, do they almost camouflage as something else in these images?
They're hard to see because they sort of camouflage as something else. What they are is the cells always have walls, of course, which divide the inside and the outside. And they're like a specialized patch of the cell wall. But unlike a neuron where that patch is bigger and sort of bulges and shows up as a black dot in a microscope, these don't. They look sort of like just the regular cell wall that you see. And this is there's another technical reason why they're hard to see, which is that if you look inside a cell, normally they're slightly separated by the intercellular medium. There's water and stuff in between the cells. And when you have a gap junction, they're actually pressed together, which they aren't in other places. Unfortunately, one of the first steps in preparing a sample for the electron microscope is sort of to suck all the water out of it, because you have to embed it in plastic and everything so you can put it in a vacuum. And so that flattens all the space between the cells. And so now they're all touching each other everywhere. So these places where they were touching that were special now look completely ordinary. And so they're just it's just a combination of the techniques of electron microscopes and staining and things that make them hard to see. Now, what people are working on is, this is the reason you might go to optical, is if you can put a little tag on the protein that makes up the gap junction, and then you can attach to that, you can have a dye that attaches to that tag, then you can see where they are. And hopefully it won't stick to the cell wall in general, it'll only stick to the certain proteins that form the gap junctions, and you can see them. And so then the problem is figuring out what cell they're on. But there, maybe you can use this expansion stuff, right? So if you can stick on the tag and you can expand it, maybe you could figure out what kind of cell it is and who it's attached to on the other side. So there are people looking at these things. It's just unfortunate it doesn't show up in the electron microscopes.
But that does sound like a pretty applicable example of combining techniques that, as you mentioned, that you can use this tagging and perhaps this expansion at the same time once these techniques start to get optimized.
Yeah, exactly. So that's why I think the field has a good future. There's a lot of things that can be done that haven't been tried yet or haven't been optimized yet. Maybe we can get this information that we're missing.
A bright future ahead for sure. And then another quick question I had is, could you explain some of the ways to our listeners that maybe the findings of this field can end up being used in other fields, for example? Like, I feel like the findings of this field they could be very applicable in things like computer science, for example, if you're maybe trying to do some sort of neuromorphic computing, or maybe even medicine, just understanding the neurons.
I can think of two examples sort of like that. One is, as you said, neuromorphic computing, because if you look at the way people build computers, they build it out of gates, and the gate maybe talks to four other things, has three inputs and one output. And they string long strings of these things together. If you look inside a computer, a typical string of gates is like 60 long. It's adding up 32-bit numbers, and each bit takes 2 gates and so on. So they end up with long strings of gates with small numbers of connections. And biology is exactly the opposite. If you look at a fly, they got these neurons. These neurons connect to hundreds of other neurons. And the networks are very shallow. You can get from any cell in the fly to any other cell in 5 hops. Or something like that, which is very different from the way people build things. And yet the fly can do visual processing enough to fly and keep stable and everything with these tiny amounts of power and these small physical sizes, using these circuits that are built in a way that's unlike any built by people. If you look at the controller for a drone, it can also keep it stable by vision and stuff, but it takes a watt. The fly takes 120 nanowatts, right? So it's a big difference. And if we understood how these computations work in the fly, maybe we could build technology that's parallels that and get some of these advantages for human design stuff. And then another one that you ask, a similar one, this is at the other end of the scale, is maybe some of the stuff we can learn with flies can affect medicine. Like I said, maybe people medicine. And I think there's a great example of that potential, which is perhaps understanding the basis of alcoholism and substance abuse. That it turns out if you take flies and you put them in a chamber, and the chamber has alcohol over here and food over here, most of the flies will just eat the food and drink the water and stuff. But a fraction of the flies will drink the alcohol until they fall to the bottom of the chamber. They literally pass out. And it's the same percentage as people, which is sort of interesting, 10% or so. But then, this is Ulrike Heberlein did this. He worked at our place. But say you take two groups of male flies. And you take one group and you put them in with females that have never mated before and are eager to mate. You take the other group and you put them in with females that have already mated and are guaranteed to reject the advances of the male flies. Then you do the alcohol test. What you find is the males that got rejected drink at several times the rate of the ones that successfully mated. And that kind of behavior, of course, is familiar to anyone, college freshman or similar things. And so maybe some of these things where the brain looks at different kinds of rewards and decides which one to pursue and things like that, that may be a mechanism that's really ancient. And we can figure out things about that by studying flies. So some of the stuff, a lot of people say, certainly my relatives say, well, what are you going to learn from the fly that you really couldn't, you can really use? But some of these things may be relevant, that a lot of biology is conserved. And it may be that some of these things we learn in a fly are actually relevant to people.
I never had heard about these alcohol experiments on the flies before, but that's super interesting that you're seeing not only the same ratios that you would see in human populations, but then you're also seeing the behavior of the flies changed and based on various reward settings.
Right. So you figure that somewhere in your brain has to be something that compares the different kind of rewards you can get and how hard they are to achieve and things like that, and decides on your course of action. And what it looks like from these experiments is that maybe that mechanism is similar in flies and people.
Yeah.
If you understood what drives flies towards addiction, maybe you can understand what drives people towards it.
Right, exactly.
And so maybe this stuff will turn out to be really useful. Maybe not, right? Maybe there are two mechanisms that are very different that just look the same. You don't know until you do the study, but maybe it will be something that could impact human medicine.
That's definitely a super interesting aspect of the research there. And I'm also excited to keep an eye on that and see the further analysis on that and see what happens as people look deeper into that. And so I don't think I have any additional specific questions for you today, but I was wondering if you had anything else that we haven't discussed yet that you'd like to mention or bring up.
We've gotten through most of the points that I thought were interesting. It's a pretty wide-ranging discussion. The take-home messages to me are really that it's a field that's still wide open. And unlike other fields, which are hard to contribute anymore to, if you want to contribute to theoretical math, you better be a really, really, really good mathematician.
Right.
If you want to contribute to the biology, there's all sorts of things that are open to people with more or less ordinary skill sets. And so I'd encourage people to take a look at biology, see what needs to get done, see if you can help. There's a lot of opportunity there, I believe.
Yeah, absolutely. Definitely lots of spaces available in biology to still make an impact or maybe discover something new. As we head towards concluding the episode, should our listeners contact you for further discussion after the episode? And if so, who should reach out to you? How should they reach out to you? And what sorts of topics would you want to be reached out to you about?
Well, probably the topics I can most likely help with would be things to do with reconstruction and connectomes and things that we discussed. The other ones, other topics, I wouldn't have any more expertise than anyone on the street. Email is probably best. They can contact me. I'm louscheffer@gmail.com, sort of exactly what you would expect. And if they have questions about connectomes or how to get into biology, those would be two areas I'd be happy to discuss.